Over the course of a human lifespan, the primary engine of the adaptive immune system structurally collapses. In a process called age-related involution, the thymus, responsible for training immune cells, loses its active epithelial meshwork, which is physically replaced by inert fat. As this scaffolding disappears, the organ's biological output fails. The production of naive T cells, the blank slate cells required to identify novel pathogens and cancer cells, grinds to a halt. Without a steady supply of these cells, the body relies on an aging pool of existing memory cells. This depletion leaves the organism vulnerable to new infections and significantly impairs its capacity for tissue repair. The physical degeneration of this single organ acts as the primary upstream driver of systemic inflammaging, the chronic, low-grade inflammation that accelerates biological aging across the entire body. Thymic integrity is maintained by thymic epithelial cells, or TECs. Their architectural function is governed by a master regulator, the transcription factor Foxn1. This chart tracks the impact of Foxn1 expression. The thymus does not degrade through a linear decline. Instead, when expression levels drop below a 50% threshold, the system hits a biological bottleneck and T-cell production plummets. Maintaining Foxn1 levels is required to prevent this collapse. Recent data reveals that this immune factor is regulated by a neuroendocrine hormone traditionally associated with the brain and social behavior, oxytocin. By reverse engineering the intracellular kinematics of this connection, we can see how oxytocin intercepts the canonical Wnt pathway to rescue Foxn1 expression and preserve immune longevity. We begin inside the nucleus of a single thymic epithelial cell. Here, the Foxn1 gene promoter sits idle, attached to a waiting left TCF transcription factor complex. Postnatal transcription of this gene relies on signals from the Wnt beta-catenin pathway. To activate Foxn1, beta-catenin must translocate from the cytoplasm, enter the nucleus, and lock into that empty left TCF complex. The availability of free beta-catenin in the cytoplasm serves as the final gatekeeper for thymic survival. In the baseline state, beta-catenin is constantly dismantled by the destruction complex. Its active engine, the kinase GSK3-beta, attaches a phosphate group to beta-catenin, routing it directly into a proteasome where it is shredded into component parts. If this destructive loop continues uninterrupted, Foxn1 transcription completely stops, and the vital thymic tissue begins to rapidly involute. The rescue process begins at the cell membrane. Circulating oxytocin arrives, locking into the oxytocin receptor, a 7-transmembrane G-protein-coupled receptor. This binding triggers protein kinase C and AKT to cascade downward toward the destruction complex. These kinases target GSK3-beta, attaching a phosphate group to its serine 9 residue. This causes the entire destruction complex to physically shatter. With the destructive loop stopped, beta-catenin begins to rapidly accumulate in the cytoplasm. The stabilized beta-catenin now enters the nucleus, binds to the left TCF complex, and drives the transcription of Foxn1. At the tissue scale, this molecular rescue translates into physical regeneration. Sustained Foxn1 allows thymic epithelial cells to rapidly divide, restabilizing the three-dimensional structure. During natural aging, however, circulating oxytocin levels drop, and the density of oxytocin receptors on the thymus decreases. Without this upstream stimulus, GSK3-beta inhibition weakens, beta-catenin is destroyed, and Foxn1 levels drop below the threshold required for organ maintenance. Thymic involution results from the progressive withdrawal of this neuroendocrine signaling. This intracellular mechanism functions as part of a wider physiological circuit. Oxytocin production in the hypothalamus is influenced by external inputs, including signals from the gut microbiome that travel to the brain via the vagus nerve. Symbiotic gut bacteria, such as Lactobacillus reuteri, can upregulate endogenous oxytocin through this pathway, directly stimulating the kinase cascade we have modeled. Leveraging this systemic circuit, through microbiome modulation or direct oxytocin administration, provides a target for physically rejuvenating the aged thymus. The immune and neuroendocrine systems operate through a hardwired molecular co-regulation. Immune longevity is an active state, requiring continuous neuroendocrine support.